Alright, I'm Daniel,
I'm Erik.
and we're going to talk to you guys about the transcriptional targets of planarian retinoblastoma associated protein 48, and especially the targets that are involved in chromatin remodeling and neoblast differentiation. So you guys already know about planarians, so I'll be brief, but they're regenerative freshwater flatworms. Their regeneration is due entirely to their neoblast population, and so that's 20% of their total cells are neoblasts. They're dispersed throughout the parenchyma. They're the only proliferating cells in asexual planarians, and they have a unique transcriptome and proteome. So they have increased expression of cell cycle genes and stem cell markers, and that's what makes them different. And
Sorry to interrupt. Maybe can we pull the curtains in the back? A little better. Thanks. Much as I love the sun. That's better. That's best. Okay.
Okay, so you guys probably know a lot about human retinoblastoma. Deletions, or the loss of a functional RB, leads to childhood eye cancer. And so normally it works as a tumor suppressor gene. So it usually binds E2F and inactivates it. And then CDKs come and phosphorylate it, and that releases E2F to go and transcribe cell cycle genes that keep the cell cycle going. So RB is a tumor suppressor in humans. In planarians, it's a little different though. E2F, the E2F homolog in planarians is repressive. So everything works the same, right? Is RB binds it and then activates it and then gets phosphorylated and lets E2F go, except E2F is a repressor. So that makes RB an oncogene instead of a tumor suppressor. So planarian RB is necessary for cell cycle entry, neoblast self-renewal, and regeneration. So what does it have to do with RBAP48, right? So RBAP48 is an associated protein with RB. It's part of the pathway. It was originally discovered because it binds to RB. And it's been found to be associated with proteins like NERD and HDAC. So NERD and HDAC are chromatin remodelers. HDAC especially is a pretty well-known one that condenses DNA based on acetylation signatures. It changes the acetylation signature, and that allows other proteins to come and condense the DNA. So it's also, in other monoorganisms, it's been implicated in chromatin remodeling. It regulates the RAS pathway in C. elegans. It's involved in epigenetics, so there's a lot of stuff that it might do, and we don't really know too much about what it actually does for sure. And the thing that we're more interested in is evidence suggests that it plays a role in the transcription of the repression of the transcription of E2F targets. So there's evidence that it works directly as a transcription factor to repress E2F target genes, and that's kind of what we're focusing on. So what's known about RB and planarians? There isn't any research at all about the in vivo mechanism, so we don't know too much about it. But we know that its expression is restricted to neoblasts, and we know that when you knock out RBAP48, that the neoblasts still get activated, but they don't differentiate. So you wound it in a knockout. You wound it. Neoblasts still go to the wound, but they get there and they don't differentiate. So there's kind of this what's going on? How does it actually affect neoblast differentiation? And that's what Erik's going to talk about.
So Daniel and I believe that RBAP48 works as a transcriptional factor. There's this big gap between, we know what RBAP48 does. It's important for neoblast differentiation, but we don't know exactly how it achieves that goal. So we think it works as a transcription factor because of these two pieces of evidence. So in a recent paper that was published where they RNA-I'd RBAP48, they found that there was changes in chromatin remodeling, but there were no accompanied changes in HDAC activity. So the researchers observed that chromatin became more condensed, but the condenser protein, which is HDAC, had no change in activity, wasn't really involved in that process. And we also know that AP48 represses E2F target genes, and so it has the ability to bind to the genome and to DNA. So we put these two pieces of evidence together, and we're trying to figure out, our broad question is how does RBAP48 regulate neoblast differentiation? And there are, maybe some other unknown factors that play into this question. But our piece of the puzzle, what we can figure out is, what are the transcriptional targets of RBAP48? And this is kind of what we can contribute to this overall question. And in order to answer this question, we need to first answer these specific questions. So first, we need to figure out where RBAP48 is located. We need to know where it is so that we can conduct our research and our experiments on those specific cells and at the specific location in the cell. We need to know when does it interact with the genome. We want to figure out when RBAP48 expression is highest, and hopefully when it's highest is that it'll be bound to the DNA, and that's when we can do some experiments on it. We need to know what genes are regulated by RBAP48, and hopefully maybe some of those genes are chromatin remodelers or epigenetic regulators, because the process of stem cell differentiation involves changes in chromatin architecture and epigenetics. And so if RBAP48 could possibly regulate some of the chromatin remodelers and epigenetic regulators, then we can see how AP48 is essential for differentiation. So this slide just kind of, again, shows the general overarching question is, well, how does AP48 regulate neoblast differentiation? And this is our goal. Our goal is to determine and to characterize the transcriptional targets of planarian RBAP48. And we're going to achieve this goal by answering these 4 questions. So why is this significant? Why is this important? So first and foremost, there currently is no research whatsoever about the in vivo mechanisms of planarian RBAP48. And if we can understand exactly how AP48 works, we can begin to understand how chromatin architecture can influence totipotency and differentiation. And also, we can gain insight into which genes or pathways are essential for planarian neoblast differentiation, because if we know that AP48 is essential for differentiation, and then those targets would also be part of those pathways that are absolutely essential for it. Also, it's been shown that the retinoblastoma pathway in general is very important for planarians and for their neoblasts. They've done studies about knockouts with RNAi for retinoblastoma, and the planarians are unable to differentiate. They're unable to regenerate. They can't regenerate new tissues. Tissue homeostasis is compromised. And so if we can add the knowledge of RBAP48 and its mechanisms to the overarching retinoblastoma pathway, we can kind of just add to the total knowledge of RB. And also, the more we know about this model of differentiation, the more we can begin to experiment with differentiation tests. So planarians right now already a very good model for stem cell biology tests, and the more we know about differentiation, the more experiments and research that can be conducted. And also, it's been shown that RBAP48 has sequence homology to a human retinoblastoma binding protein 4. So it would be really interesting, because we know that there's sequence homology, but is there maybe a folding homology or maybe a functional homology, and that could be good for future studies if we want to do studies on RBBP4, we could do it in planarians, and also just for an evolutionary sense is how closely related are we by sequence. So Daniel's going to talk about our experiment.
So how do we go about actually, answering these questions, right? So we want to find out when and where RBAP48 is present. And so we're going to do that. First, we're going to wound, because we know that RBAP48 is involved in the regeneration process. So we wound them, and then we're going to do a whole mount in situ hybridization. So that's going to give us the mRNA expression profile. Then we're going to do an immunohistochemistry assay, and these are all being done at different time periods after. So one will be done after 1 day, after 2 days, after 3 days. And we're going to find the most fluorescence, and that's going to be the time period that we use for our following experiments. And so then we're going to do a protein localization assay. So we want to know whether or not it's in the nucleus or the cytoplasm. So the way that works is we have 3 markers, right? We have known nucleus markers, known cytoplasm markers, and then we light up RBAP48 with a 3rd color. And we overlap those 3 images and see which marker RBAP48 overlaps with the best. And then, so for controls, we're doing Western blots, so we want to make sure that that the antibody we're using for the immunohistochemistry binds selectively. And then for the in situ, we want to use a scrambled probe to see that also that it binds selectively, that it's not going to bind if the RNA isn't there. Okay, and so we know that RBAP48 expression is limited to neoblasts, so we're going to use FACS to take out the neoblasts, and we're going to do that based on these 2 inherent neoblast characteristics. So neoblasts have increased euchromatin, so their DNA is less condensed, and they have decreased cytoplasmic volume. And so that's how we're going to use FACS, or that's the basis for which FACS is going to separate the neoblast out. Okay, and so this is kind of the bulk of the experiment, right? Is the ChIP-seq. So we want to know what genes are actually being regulated by RBAP48. So the way ChIP-seq works is we're going to cross-link the proteins to the DNA. Hopefully at this time when we cross-link it, some of the RBAP48 is going to be bound to the DNA. So we cross-link it, then we digest it, and we basically put it through an affinity column. And so we have beads with antibodies that are going to bind to RBAP48, and the DNA is going to be cross-linked, and all the DNA that isn't attached is going to flow through, and then we're going to reverse the cross-links, elute the DNA that was bound to RBAP48, and sequence it. And so now we have this kind of small region of DNA that came out that was bound to RBAP48, and so we want to know what gene is that actually a part of. So we put it in the Planarian genome browser, and so we get the whole gene sequence, and then with that gene sequence, we're going to blast that and find what kind of homology it has to other organisms. And so RB in humans has, I think, 2,000 ChIP-seq transcriptional targets, so we're kind of expecting a lot of targets, so we need to rank them based on what we're interested in. And so what we're interested in here is genes involved in chromatin remodeling and epigenetics. And so we're interested in this because we think that this is the mechanism by which RBAP48 regulates differentiation. And so we're also interested in genes that have homology to human genes. Okay, and so how do we confirm that the targets that came out of ChIP-seq actually are being regulated by RBAP48, right? So we're going to do RNAi followed by QRT-PCR. So we knock out RBAP48, and then we're going to quantify the amount of expression of mRNA of RBAP48 that's there, and it should be changed, right? So we just want to see that if we knock it out, that there's some difference in mRNA expressions. And so the control for this is going to be Western blots. So we want to make sure that RBAP48 actually is knocked out. So we're going to do a Western blot to do that. Okay, so this is our overview, just as a big picture. So we want to know when and where is RBAP48 expressed, right? So we wound, and then we do a home out in situ and immunohistochemistry at different times. We look for the most fluorescence because that's where the most RBAP48 is going to be. We use that time period for finding out what genes are regulated by RBAP48, and we're going to do that. We're going to use FACS to separate the neoblasts, and then we're going to do ChIP-seq to find the actual genes that are being regulated. Then we get, to find the functionality, we get the whole gene using the Planarian genome browser, and we put that whole gene into BLAST to find what sort of functions or what sort of homologies this gene has to other organisms. And we confirm our results by doing an RNAi and quantifying the amount of expression changes by QRT-PCR.
All right. So for our wounding experiment, what we can expect, what we hypothesize will happen, is that the neoblasts will be the only cells expressing RBAP48. And other research has shown that this is the case. We also expect that peak expression will occur 2 to 3 days after wounding. And we can come to this hypothesis because after about 2 to 3 days after wounding, the blastema forms along the wounding site, and neoblasts will line the basement of the blastema, and they will begin to proliferate and differentiate. So when they're differentiated because they want to replace those missing cells and those missing tissues, so RBAP48 expression should be highest there because of the blastema. And also we believe that AP48 expression will be localized to the nucleus because it acts as a transcription factor. It's involved with this chromatin remodeling process, so that's where it should be localized. So for our facts, neoblasts will be separated based on euchromatin and low cytoplasmic volume. So we're using 2 different types of stains. The first stain is a nucleus stain. It's called the Host stain, and it stains for condensed chromatin. So because neoblasts have euchromatin, like their totipone, a lot of their genes are open, they're not going to have a very high Host stain, so they have a low Host stain. And then also because their chromatin are so open, they have a larger nucleus and they have a lower cytoplasmic volume. So there's a stain that you can use called calcium AM, which stands for high cytoplasmic volume. So we should have a low cytoplasmic volume for our neoblasts. So the 2 low signals will correspond to our neoblasts. And so we'll use those to separate them out. So for ChIP-seq, this is our big experiment. Our hypothesis is that RBAP48 will be cross-linked to the DNA. We know that it has the ability to associate with DNA, and because it acts as a transcription factor during the times that we are wounding, it should be cross-linked to the DNA. And then we're hoping to get, as Daniel said, a lot of hits, a lot of target hits. So a lot of these prospective genes will hopefully come out of ChIP-seq. But we also have to think about what if nothing comes up? Like, what if nothing comes out of ChIP-seq? So we could do a protein-protein interaction study like co-IP. So, for example, maybe AP48 is associated with like a DNA binding protein, and then those two proteins as a complex go to the DNA and then transcribe certain differentiation genes, right? So it could be something like that. Or what we could also do is we could do RNAi against AP48 and analyze the transcriptome via RNA-seq. So we can sequence the RNA, and we'll be able to see the direct and indirect regulation that AP48 has. So we'll compare that to a control planarine, and so we'll be able to see all the direct and also the downstream effects of AP48 missing. So the planarine genome browser, our hypothesis is that our prospective targets should include chromatin remodelers and epigenetic regulators, right? Because, like we said before, neoblast differentiation is in general controlled by chromatin architecture, right? Certain genes need to be closed off because the differentiated cell is not going to need those genes anymore, and certain genes need to be left open because the differentiated cell will need those genes, right? So chromatin architecture changes, right? And that's done by chromatin remodelers. And also epigenetics, the methylation patterns of certain genes, to keep them open is also really important in differentiation. So we're hoping that because we know that AP48 is essential for neoblast differentiation, that some of these targets may include these types of genes. And we're going to know that because of the planarine genome browser will give us annotated depictions of the genes, and also by BLAST will be able to tell us what other organisms have similar genetic sequences. And so we also need to play devil's advocate here. It's possible that none of the targets will be involved in these processes. They won't be involved in differentiation. So some possible other scenarios is that maybe these genes are involved with cell cycle regulation, or maybe they're involved in signaling cascades that have an indirect regulation on neoblast differentiation. But still, even if we do get some hits, and even if they're not what we really expect, it'll still give us some insight as to the total function of RBAP48. So we're going to confirm our procedures using RNAis and QRT-PCR, as Daniel explained. So after RNAi knockdown, RBAP functioning should be diminished, should be much lower. And so our QRT-PCR then will be able to show us what are the changes in the target genes. So we're going to do QRT-PCR against the target genes that we're interested in. We're going to check out their mRNA expression. So hopefully, let's say, for example, if AP48 upregulates gene A, right? But when we knock it down, that means that gene A will also be downregulated. And so we'll be able to compare that to a control, and hopefully it'll show some differences that we'll be able to analyze. And then we can also say that if expression levels don't change, then maybe RBAP48 isn't actually regulating these target genes. There might be some other protein. So what are our future research opportunities? If our research proposal was funded and it went well and it was accurate and it was all fun and dandy, so the next step would be to begin to characterize the functions and mechanisms of the ChIP-seq target genes. So the genes that RBAP48 transcribes, well, if they're really essential for differentiation, right, well, what are their functions? What are their mechanisms? How can we begin to learn about those? And that'll kind of get us closer and closer to the target of, or to the goal of, well, how is differentiation achieved? We can begin to study the regulation of RBAP48 in relation to wounding and tissue homeostasis. So when a planarian is wounded, what are the signals that tell RBAP48 to begin to be active? And when the cell is done dividing and the wound has been healed, what are the signals that tell RBAP48 to shut off? Also, tissue homeostasis is essential, and there's kind of just like a slow turnover of neoblasts into differentiated cells, and what controls the slow process or the slow turnover of RBAP48. So that would be something to study as well. And also we could do a ChIP-seq for AP48 and other model organisms to see if they have a functional homology to what planarians have. Cool. Daniel's going to close it out.
All right. So why should you give us your money, right? So there's no research being done on the in vivo mechanism of RBAP48, and our research kind of marks the first step in figuring out how this gene is, how this gene plays into the what piece of the puzzle this gene is for regulating neoblast differentiation. And so by finding out the mechanism of how this gene works, maybe we can begin to understand how chromatin remodeling in general works to affect differentiation, which I think is one of the really big questions. One of the really big questions of our time. Yeah.
Cool. Thank you.
Questions?
I have a question.
Yeah.
So, a lot of people would probably criticize this study because this protein functions like almost opposite as it does in vertebrates. This is an oncogene. What would you say to someone who has that criticism of why this is important?
I mean, even if it's opposite, it could still shed light on the process of how it works. I mean, it's obviously pretty similar. There's sequence homology. I mean, I'm not sure if I understand exactly what you're asking, but I think that it's so important.
So yeah, RB, the retinoblastoma protein, in relation to E2F is opposite. And in fact, actually, RB is similar. It's just E2F is only the repressive homolog. So E2F is the part that's kind of opposite. The retinoblastoma is opposite because it's needed for cell cycle entry. But we're more focused on RBAP48, its associated protein. And it's really not super known how RBBP4, which is the human homolog, really acts in humans or in other vertebrates. So I guess you could kind of criticize it a little bit about because RB is opposite, but we're kind of looking at a side pathway of retinoblastoma.
Is the sort of following up on that? Is the interaction between RBAP48 and the planarian RB similar to the interaction between the vertebrate RBBP4 and RB? Is that that nature of the interaction the same?
That's a good question.
I don't think we know. I mean, no, there hasn't been anything about this particular protein's mechanism of vivo. So.
Okay, but they know that it binds to planarian RB. Is that true? Okay.
They don't know exactly how those interactions work.
Okay.
But that it's associated.
Right. Because that might influence whether this is relevant or not to understanding how things work in the vertebrate.
Yeah.
System.
And even so, even if it's not homologous to vertebrates, I was talking to Andrew the other day, and he's like, well, everyone's kind of like talking about how this will be applied to medical uses or to vertebrate uses, and it's like vertebrates are cool, but so are invertebrates. We can learn a lot from invertebrates. You can't just push them aside because they're opposite.
Right. Sometimes this may not have direct correspondence in terms of individual genes and individual pathways, but oftentimes they're conceptual kinds of information that you gain from studying invertebrates, even if sort of the individual molecules don't show functional homology.
And it may be easier to study those concepts in vertebrates as compared to
right.
Or
okay, so let's see. So going back, you said something that so RB is a tumor suppressor in vertebrates, and you said it's an oncogene in planarians. That's been demonstrated that it actually functions as an oncogene, or just because it has the opposite effect on E2F?
It's more because of the opposite effect on E2F.
I think it's been demonstrated. What they did is they knocked it out. They knocked out RB, and they saw decreased mitosis markers. I think.
No, it was the opposite. Daniel.
That's
They
the
knocked
opposite.
it. They knocked
The
it out.
opposite.
No wait.
No.
How would you demonstrate something as an oncogene?
You could overexpress it and see if there's increased proliferation.
Right.
Yeah.
I don't think they did that.
Okay.
They haven't done that. They've done RNA against it.
Yeah.
Right.
So I'm trying to figure out if that's
Right. So the other would be to overexpress and see if you get hyperproliferation or unregulated proliferation.
yeah, because RNA is a loss of function to see what's necessary.
Right. Exactly. Okay. Cool. I mean, you would if it looks like it's necessary for cell cycle progression, that's a reasonable hypothesis. Absolutely. That if you overexpress it, you should get unregulated.
Yeah.
But I'm just curious if that had been demonstrated. Okay. Let's see. So it's homology to RBBP4, the vertebrate homolog. So, I don't know if you looked at the sequence. Are there any sort of known conserved functional domains like protein-protein binding domains or DNA binding domains that might help you sort of figure out what sequence it might interact with, or
Yes.
what other proteins it might interact with?
Yeah, so it has a sequence. It's called a WD40 repeat.
Mm-hmm.
I don't remember exactly what a WD40 repeat does. It was,
I think it might just be a name.
yeah. Do you know what?
I don't know what they do.
I mean, I can look it up real quick.
No, that's fine.
But it has, so there's a specific motif called the WD40 repeat, and
Is it involved in DNA binding? Or?
I think we actually looked it up.
Okay.
We did, and I think it was. I don't think it said anything.
Right. Because one thing you could look for is if
Yeah. So,
there's any clues as to what RBBP4 binds to, what genes it binds to, or even what sequence it binds to. You might be looking to see in your putative targets if there are some similar sort of sequences to which your protein might bind. Okay, so that could be part of sort of what you'd be looking for, because you're going to get potentially a lot of hits in your ChIP-seq,
Yeah.
right?
yeah.
Yeah.
but hopefully we have a lot of undergraduates to do it for us.
Okay. One other question. So the facts. So you say you're going to do the facts before ChIP-seq. Can you explain that a little bit? Why do you want to purify neoblasts to do ChIP-seq?
Well, because we're interested in how RBAP48 regulates neoblast differentiation, right? And with the chromatin architecture and stuff, right? So we want to focus in on neoblasts because those are the only actively dividing cells in the planarian. And so, in order to do that, we're going to need a population of neoblasts so that we can isolate RBAP48, like active RBAP48. And then also our immunohistochemistry and our in situ hybridization studies are going to hopefully show that all these neoblasts that are lining the blastema are lit up because they're expressing RBAP48, right? And maybe the differentiated cells, the adult cells in the planarian aren't expressing it. And so if that's the case, then we're going to need those cells that are expressing RBAP48.
So that's a fact. It's been shown that RBAP48 is only expressed in neoblasts.
Okay. And then the RNAi. I mean, actually, I know this because I've talked to you guys, but explain how that's actually done in planaria.
Yeah. I think what they did in the papers I read is they fed it to them. They I think it was in other worms, right? Or not?
So what they did was they created these RNAi in E. coli, and then they mixed the E. coli in with their food, and so they would have like a beef liver paste type of thing, and they would feed it to them, and they would literally eat the RNAi because it's in the bacteria.
So they're just putting a transgene in the plasmid or something, expressing high levels of the whatever it is, scorderpin RNA, whatever it is.
Yeah.
And they just gobble up the bacteria.
And then they digest
Cool.
it.
So if you're going to be doing that, and then looking for the effects on expression, could you look, because and part of your idea is that this protein's involved in neoblast differentiation, and you're knocking them down. And presumably, if they're required for neoblast differentiation, they're then necessary for full regeneration. Could you look at these a subset of the individuals, and look and see if you're knocking down RBAP48, what the consequences are to their regeneration?
Yeah, so we actually know the answer already. So in the paper that they did that, they analyzed the neoblasts at the site of the wound, and the neoblasts, because they failed to differentiate, they started to apoptose. And the planarians, after about 40 days, all of them were dead. So
the neoblasts migrate okay, but they don't differentiate.
Exactly. So they're stimulated to migrate to the site of the wounding, but once they get there, they can't differentiate into the correct cell type, and they apoptose.
And one of the other things, I think they maintained they're still neoblasts, so they did marker
Right.
experience.
It might be useful though to at least have a small subset to partially to confirm that your RNAi, an additional sort of confirmation your RNAi is working in the way that you expect, is to have maybe a small population, that you confirm that in fact you're inhibiting regeneration. And so that the changes in gene expression are, in your experiment, being correlated directly to a loss of regenerative capability.
Yeah, that's a good point.
Yeah. Cool. Any other questions?
Yeah.
This is kind of a much more softball question, but I was just going to ask just a vague idea, ballpark of what kind of timeline we'd be talking about for this experiment. Any concept of that?
I mean, a couple years.
Yeah.
Easily.
Millions of dollars.
This is.
That's a good question.
Yeah, no, it is. It's probably a few years. It's a lot.
Cool. Thanks, guys.
Any other questions?
So, do you want us to save this onto your desktop or something?
Oh,
Do
yeah,
you
that'd be
want a copy
great.
of it?
Yeah.
We'll just there now. I mean, I can put it there now. Just do it one of these.
Just drag it over there. Yeah.
Cool. All right. Thank you.
Thank you. It's a mess.
Do you want to inject it? Do you want to inject it?
Kayla, do you guys need my computer or?
Oh, Jeff. Okay.
Cool.
Thank you.
Sure.
Oh, yeah.